Welcome back to the Deep Dive. Today, we are tackling what is, I think, the single most frustrating problem in modern medicine, particularly in targeted cancer therapy. It's this endless evolutionary arms race between our best drugs and the disease itself.
It is. We develop a precise weapon, we use it, it works for a bit, and then boom, the tumor evolves resistance and escapes. We're always one step behind.
And that cycle, that constant failure where we're always trying to figure out yesterday's mutation, is exactly what our source material today calls reverse engineering resistance.
Right, it's completely reactive. We have to wait for the treatment to fail, for the tumor to tell us how it beat us, and only then can we devise the next step. But that next step, it just selects for the next resistance mechanism.
So today, we're diving into a really radical shift in that strategy, something called forward engineering tumor evolution.
It sounds like science fiction, doesn't it?
It really does. So our mission for you, the listener, is to really get to the bottom of how scientists are proposing to seize control of a tumor's evolutionary path. They're using something called selection gene drives, or SGDs.
Yeah, this is a system that uses these genetic switches and small molecules to completely invert the evolutionary pressure. The goal is to make the cancer cells more treatable no matter what genetic chaos is already in there.
Okay, so to really understand how big a deal this is, we have to start with the problem itself, that evolutionary arms race. Let's really dig into why the conventional approach fails so consistently.
The foundational issue is something we call tumor heterogeneity. We used to think of tumors as, a uniform blob of identical cells.
But they're not. They're more like a chaotic ecosystem.
Exactly. Even before you start any treatment, that tumor contains tiny pre-existing subclones. These are just a few rare cells that, by pure chance, already have a mutation that will let them survive the drug you're about to use.
So when you hit that tumor with a targeted therapy, a conventional monotherapy, you're not just killing cancer cells. You're actually helping. You're actually helping the resistance.
You're doing exactly what the resistance needs. The drug is this powerful evolutionary filter. It wipes out, say, 99% of the sensitive cells, which sounds great. But in doing so, you've just cleared out all the competition for those few pre-existing resistant cells.
You've given them a wide open field to grow in.
An uninhibited environment. It's like you've weeded a garden but left the most invasive species behind to just take over everything.
And the most, I think, painful and well-known example of this is in non-small cell lung cancer, NSCLC, when it's driven by mutations in the EGFR gene.
That is the canonical story of this failure. A patient with EGFR mutant lung cancer gets a first-line drug, a TKI like erlotinib, and for a while, it's amazing. The tumor shrinks beautifully.
For a while.
For a while. But soon enough, resistance pops up, and it's almost always driven by a specific new mutation called T790M. So what did oncology do? Well, they reverse engineered the problem and developed a second-generation drug, osimertinib, specifically to hit T790M.
And that works until the tumor plays its next card.
Right. Now, osimertinib provides the selective pressure for a tertiary resistance to emerge, and that's usually another mutation called C797S. We are always one drug, one generation behind.
The tumor always has another move. So if combination therapies are often too toxic and this one-by-one sequential approach just guarantees relapse, we need something completely new.
We need to control the selective process itself, and that's where this SGD system comes in. It's designed to completely flip that evolutionary script.
So how does it work? I understand it's a dual switch mechanism that gets delivered right into the tumor.
That's right. The SGD approach modifies the cancer cells in situ, so right there in the body. The whole circuit has two parts, two switches, but they're integrated into a single genetic package.
Let's start with switch 1, because this is the really counterintuitive part.
It is. Switch 1 is essentially a synthetic resistance gene. We're actually modifying a fraction of the tumor cells to give them a temporary resistance.
Oh, wait, hold on. We're intentionally giving cancer cells resistance.
Yeah.
Why on earth would we do that?
Because we want to choose the winner of the evolutionary race. When we start the treatment, the patient gets two things: the targeted therapy, let's say osimertinib, and a small molecule that activates our switch.
Okay.
So what happens? The normal, sensitive cancer cells die from the osimertinib. The naturally resistant cells, those C797S mutants, they start to grow. But our engineered SGD cells, protected by our controllable switch 1, they grow even faster.
I see. So the goal is to make the population we control the new dominant group in the tumor. We want our engineered cells to totally outcompete those dangerous, naturally occurring resistant cells.
Precisely. We are guiding the tumor to evolve toward a state that we designed. We're overwhelming the native resistance simply through abundance. Our engineered cells might even be a little less fit, but they start out orders of magnitude more numerous than the natural resistance, so they just win the race.
And once we've established this new dominant population that we control, then switch 2 comes in for the kill.
Exactly. Switch 2 is the therapeutic payload, a suicide gene, basically. And this gene, which is part of the same genetic package, activates a diffusible therapeutic agent. And this is where the really crucial idea of the bystander effect comes into play.
Okay, so what does that mean, bystander effect?
It means that once switch 2 is turned on, the drug it activates kills not just the engineered SGD cells that are making it, but also every single neighboring unmodified cell.
Yeah.
That includes those nasty native-resistant subclones that we outcompeted in the first phase.
So we're turning our own engineered cells into little drug factories to poison the entire tumor mass from the inside out.
That's the idea.
But hang on. This sounds a lot like something called GDAPT, gene-directed enzyme pro-drug therapy, which has, historically struggled. The big problem has always been delivery. How do you get the gene into enough cells in the first place for it to work?
That is the million-dollar question.
Yeah.
And it's the critical challenge that SGD was designed to overcome. The answer really lies in the rigorous computer modeling the researchers did before they even picked up a pipette.
Right, which brings us to the model-informed design. It's fascinating that they basically tried to predict how it would fail using simulations first. What did those models tell them?
They use these really sophisticated stochastic models, which are complex algorithms that simulate cell growth and selection over time. And these simulations gave them 4 key design criteria that just fundamentally change the math for gene delivery.
Okay, let's focus on that first one because it sounds like the most revolutionary bit for gene therapy as a whole.
It really is. The primary finding was that the selection pressure from switch 1 can completely compensate for inefficient gene delivery. The model predicted that you can achieve evolutionary control even if you only successfully modify an incredibly low percentage of cells.
How low?
Potentially less than 1%.
Less than 1%. That's a game changer. I mean, for a normal suicide gene therapy to work, you need really high efficiency, right? Like 70, 80%. Otherwise, too many cancer cells just escape.
Exactly. But here, that selection step means you only need to hit a tiny fraction of cells, and then evolution does the hard work for you, amplifying them until they dominate the whole tumor. It just lowers the bar for clinical translation enormously.
So you can focus on making your delivery vehicle safe and targeted instead of just brutally efficient.
Precisely. And a related finding from the model was about competition. Even if your engineered cells are a little less fit than the native resistant ones, they still win. Because even at 1% delivery, your SGD cells are still way, way more abundant than the naturally occurring resistant cells, which might only be, say, 1 in a million at the start. Abundance beats a minor fitness disadvantage when that selective pressure is on.
Okay, so the model solves the delivery and competition problems. What about the physical layout of the tumor, the spatial problem?
They modeled that too, using what are called spatial agent-based models. And those simulations confirmed that switch 2 absolutely must have a diffuse bystander effect. The kill radius of the therapeutic agent has to be wide enough to reach and kill those neighboring unmodified cells.
Which is what pushed them toward a prodrug system, where the activated drug can just seep outwards.
It steered them directly to it. It was a crucial design insight.
This is a perfect segue to the actual prototypes. How did they actually engineer Switch 1? How did they build that remote-controlled suit of armor?
So they engineered what's called an inducible kinase analog. They took the target protein, like the EGFR kinase domain, and they fused it with 2 key things. First, a resistance mutation, like C797SS.
The one that beats osimertinib.
Right. And second, they fused it to a little molecular component called the FKBP12 domain. This domain is a synthetic switch that forces proteins to pair up to dimerize.
And that dimerization is what a lot of these cancer-causing proteins need to be permanently on.
Exactly. But this switch needs a key, and that key is a small molecule called AP2187. When the patient takes this molecule, it binds to that FKBP domain and forces the engineered resistance proteins to pair up. This creates this powerful, unregulated on signal that rescues the cell from the TKI treatment.
Very neat molecular locking key system. So what about Switch 2, the diffused drug factory?
For that, they chose a classic GDPT system that perfectly matched their spatial criteria from the model. the enzyme cytosine deaminase.
Okay, what does that do?
Cytosine deaminase is an enzyme that takes an inert, harmless prodrug called 5-fluorocytosine, or 5-FC, and it converts it into the potent chemotherapy drug 5-fluorosil, or 5-FU.
And 5-FC is already an approved antifungal drug, right? That's a huge plus for getting this into the clinic eventually.
It's a massive advantage. And the 5-FU that gets produced by the engineered cell is highly diffusible. It leaks out and poisons the whole surrounding area, satisfying that critical need for a strong, wide-reaching bystander effect.
So how did the lab tests, the in vitro tests, prove that you really needed both steps?
Well, they set up a dish with a mix of cells. Sensitive cells, a tiny fraction of native resistant cells, about 0.5%, and then about 5% of their engineered SGD cells.
Yeah.
In the switch 1 phase, so TKI plus the dimerizer key, the SGD cell successfully took over, overwhelming and suppressing the growth of the native resistance. Then, when they switched to phase 2 TKI plus the 5-FC prodrug, the entire population just collapsed. It was wiped out.
And what if they skipped the first step?
Critically, if they skipped switch 1 and just went straight to switch 2, the native resistant population was not cleared. It survived and grew. This proves that selection phase isn't just helpful, it's absolutely mandatory for the whole system to work.
Okay, so that's a great proof of concept against one specific known resistance location. But real human cancers are just genetically chaotic. Which brings us to the really critical question. Can this thing handle the full-blown genetic mess of a real tumor?
That's exactly what the researchers had to prove next. They knew the system had to be completely agnostic to the specific type of resistance. So they put their SGD construct through 3 incredibly tough genetic stress tests.
Let's hear about the first one.
First, they tested it against resistance caused by what are called bypass oncogenes. Think of these as the cancer cell turning on a whole new engine when you shut the first one down. It activates a parallel signaling pathway.
So it makes the original targeted drug totally useless.
Completely useless. They tested resistance from genes like ALK, BRAF, and TRKA, and in every single case, the SGD system successfully eliminated these resistant populations.
Okay, so that confirms that even if the resistance is coming from a completely different pathway, the selection pressure from switch 1 is still strong enough to let the engineered cells win. What was test number 2?
The second test was all about on-site, internal chaos. They tested the SGD system against a massive genetic library containing 2,717 different single amino acid changes in the EGFR protein itself.
2,700. That's a level of diversity you'd probably never even see in a single patient's tumor.
Far beyond it. And yet when they applied the SGD protocol, they achieved complete eradication. It proved the system doesn't care about the specific nature of the on-target resistance.
That is an incredible level of complexity to overcome. And then they went for the ultimate stress test.
The final test, the InTrans challenge. They used a genome-wide CRISPR knockout library representing an unbelievable 76,441 different genetic variants. These are knockouts in genes all over the genome tumor suppressors, cell cycle regulators.
Genes that have nothing to do with the EGFR pathway directly.
Nothing. It's the definition of genomic chaos. And even against this population, the SGD selection, followed by the switch 2 payload, achieved complete eradication.
That's incredibly compelling evidence. It really suggests the system can handle the whole spectrum of genetic chaos without needing to know the escape plan in advance.
And its robustness is matched by its modularity. They showed it wasn't just for EGFR, swapping it out to work with a RET inhibitor in thyroid cancer cells. And, maybe most importantly, they also swapped out the drug factory switch, 2 for a completely different killing mechanism. Immune activity.
An immune switch.
Right.
How do you get that required diffuse bystander effect with the immune system?
It's very clever. They engineered the cancer cells to express a surface marker called CD19. Then they added T cells and a drug called blinatumomab, which is a T cell engager. It physically links the T cells to the engineered CD19 positive SGD cells.
So the T cells are forced to attack the engineered cells.
Right. And when the T cells kill those SGD cells, they also release these non-specific cytotoxic signals into the local area. It's a process mediated by something called FARSA signaling.
So the T cells become the local poison factory. Instead of 5-FU diffusing out, it's this non-specific kill signal that spreads to the unmodified resistant neighbors.
Exactly. It's so important because it shows you can plug and play different therapeutic mechanisms into this framework. It's not just limited to one type of payload.
So let's bring this all together and move to the in vivo data. All this modeling and lab work led to an optimized treatment schedule that was apparently critical for success in mice.
It really was. The computer models had predicted something subtle, that it was best to keep switch 1 activated, to keep giving that dimerizer molecule for a short period of time after you start switch 2.
And why does that little bit of overlap matter so much?
Because the production of the 5-FU poison relies entirely on the engineered SGD cells being alive and healthy. If you turn off their protective switch 1 too early, the main targeted therapy just kills them off. You shut down your drug factory before it's had a chance to do its job.
So by keeping that overlap, you keep the factories running longer, maximizing the local poison production.
You maximize it. And in the mouse models, that subtle scheduling change, which came straight from the computer model, dramatically prolonged the time to progression.
Which brings us to the final long-term survival experiment. What did that show?
The results were just stark. They used the optimized schedule in mice with a low, clinically relevant frequency of resistant cells. In the control group, the mice just getting the standard targeted therapy, you saw the classic pattern. Tumors shrink, and then they inevitably relapse. The median survival was 57 days.
And in the gene drive arm?
In the gene drive arm, where they started with only 10% of the tumor cells being SGD cells, nearly every single mouse, 11 out of 12, achieved a durable response. They had no palpable tumors at the end of the study.
Wow.
It was a massive, highly statistically significant extension of survival, a p-value of 2 × 10 to the -6. This level of efficacy against known evolving resistance in this kind of model is, well, it's pretty much unprecedented.
So the big takeaway here is this incredible conceptual shift. We're moving from playing defense, from chasing resistance, to proactively programming the tumor's evolution toward a vulnerability that we control completely.
That's the whole story. We are not just trying to find a better drug, we are using evolution itself as the therapeutic tool.
So looking forward, what are the hurdles? I imagine getting this into human trials still comes down to delivery, right? Especially to distant metastases.
That is always the classic challenge for gene therapies, no question. But as we talked about, SGD has this unique advantage. Because the whole system leverages selection to amplify that initial small population, you don't need 90% efficiency.
You just need to get a foot in the door. Less than 1%.
You get a foot in the door. And this allows researchers to prioritize safety and tumor selectivity in their delivery vectors instead of just raw power. It's a unique benefit that might just allow this whole approach to bypass some of those long-standing translational barriers.
It's incredible. It's turning cancer's greatest strength, its own evolutionary dynamic, into its ultimate undoing.
It really forces a new way of thinking. We're moving from being passive observers of mutation to active programmers of therapeutic vulnerability.
Which leaves us with a final provocative thought for you to consider. This research shows we can engineer vulnerability directly into cancer. As we move from passively observing a tumor's evolution to actively programming it inside a patient's body, what ethical or technological considerations beyond just safe gene delivery become the most pressing for the field of oncology? How might this ability change the very nature of patient consent when we're intentionally manipulating a disease's evolution?
Thank you for joining us for this deep dive into what could be the future of anti-cancer therapy. We hope this is giving you a fresh perspective on this exciting new framework.